I just wanted to thank Lou Bloom, who arranged for this, for inviting our speaker this month. Any of you that have some suggestions of folks to participate in our webinar series, please feel free to contact us, and we'll be more than glad to consider that. First of all, I just want to mention you're welcome to ask questions. There's a chat box, and we'll be having questions at the end of the presentation and possibly in the middle of it. So at any time, feel free to enter your questions you might have. We also have an opportunity at the end. You can do what's called raise your hand and we can open up the line for a verbal question. So I'd like now to introduce our presenter for this month. Dr. Reavie is a Senior Research Associate and Assistant Initiative Director for the Natural Resources Research Institute at the University of Minnesota Duluth. Dr. Reavie and his research team pursue research and applied aquatic studies on freshwater ecosystems, evaluating water quality issues. Routine work focuses on the use of algae as indicators of environmental changes. The use of these microscopic primary producers as indicators of change is especially powerful because it can predict changes at the base of the food web before more widespread impacts are realized. In his presentation today, Dr. Reavie will discuss case examples of recent eutrophication and climate change to illustrate how their research team uses these microscopic biological tools in the Laurentian Great Lakes. Also, and we're glad he's focusing a little bit on this, the quality assurance and quality control techniques will be discussed to ensure precision in taxonomy and inferred stressor impacts. I'll now turn it over to you, Ewan, who will be presenting on the topic of very tiny things, provide an early warning system for the Great Lakes. Ewan, now your turn. Thank you so much for presenting for us.
All right, thank you. And let me go up here. I'm going to turn on my laser pointer. Hopefully people can see that. Okay. Thanks for this opportunity. I wanted to give a presentation, a bit of a summary of a lot of the work that my research team does here at the University of Minnesota Duluth. A lot of it in collaboration with the US EPA Glenpo office and a number of other agencies that you can see on this opening slide here. I mean, I'm gonna go through some projects that have been funded by multiple agencies. This concept of early warning is becoming a bit more popular in recent years. We have a lot of contemporary systems in place to provide early warnings, such as that we get from weather systems to inform us about what's coming off the oceans and so on. I have a bit of my own opinion about what this term early warning actually means. I mean, if you go to the literature and you look it up in a scientific context, often early warning is referring to signals that we get from environmental systems. I'm a part paleoecologist, and if you, in the literature, you'll sometimes see long-term historical records, sometimes going back millions of years, looking at early warning signals. I consider early warning much more of an anthropomorphic term in that, you know, a million years ago, no one was actually warned about anything. The way I consider it, at least in this talk, is early warning is something that is new and upcoming and is based on recent records that are telling us about a potential future condition. Okay, so that's my perspective. I don't necessarily think everyone would agree with that, but that's going to be how I focus on this in this talk today. This is a paper from about 10 years ago, so it isn't a brand new term. This is something that is becoming realized, that there are records out there that can tell us about possible critical transitions in environmental systems that can lead to unwanted states in the future. All right, so that's where I'm going with this. So we have a lot of goals for our monitoring programs and our paleolimnological work. Again, I'm just going to focus on this idea of future unwanted states, and can we warn about where things are in the future so that we can inform water managers. All right, so this is an arbitrary organism, if we are concerned about something like schmarfs, whatever that is, and we have a long-term monitoring program, we can see, first of all, we want to be able to characterize our natural variability in the system. It'll waver a bit. But then if we come into modern times, as shown in the blue on the right, obviously something is going on here. If we're seeing a decline in these random organisms, this would suggest a warning that something is taking place here if we actually have these data. My group works on these so-called tiny things. We have a significant Great Lakes program where we are working on algae as indicators of environmental condition. And algae are these ideal indicators of condition because they are very low in the food web. They tend to be first responders to water quality changes. If the system condition changes, such as a change in pH or nutrient concentration, the algae community will respond quite quickly, and so they are good indicators of change well before things higher in the food web like fish and so on would respond. They are obviously an important component of the food web. They themselves can be a problem. If you work on Lake Erie, you're well aware of this, that harmful algal blooms appears to be an increasing issue in aquatic systems in North America. And they're not only taxonomically diverse, but they're diverse in the ways that they can be used. We can look at all sorts of different types of indicators based on the algal assemblage that we collect, whether it's diversity records or density, like cells per mil, or actually quantity, volumetric quantity in a sample, functional types, and so on. And I'm going to talk a bit about these methods later and how we distill complex algal data into indicators that are useful to managers. So let's look at a particular algal species. This is a diatom. I'm going to be focusing on diatoms for the most part here as a case example. This is a species called Cyclostephanos dubius. It's a well-known high nutrient indicator in freshwater aquatic systems. And so if we were to have long-term data that looks something like this might in itself be an indicator of an increasing problem, potentially eutrophication, increase in phosphorus load, and so on, things related to nutrient pollution in a system. We generally don't go as simple as single species indicators. I provide that just as an example. Something that uses a bit more of the complexity of these algae data might be an examination of the diversity of the assemblage that we find. If we see a sudden decline in the diversity in recent times, that might suggest there's a problem in the system, some stressor that is preventing a lot of the more sensitive species from existing. Diversity is a pretty coarse indicator of change. We've actually seen cases where stress has increased diversity, but we have plenty of options available to us. When it comes to algae, like I say, there are all kinds of tools that we can derive from the algal assemblage or scores. So I'm going to talk a bit about diatom inferred phosphorus and how we use the algae to infer water quality variables. But there are all sorts of biological indicators or IBIs out there as well, some for the Great Lakes. There are dozens of them worldwide that take the algae data in some way and distill it down into something that is informative about environmental stress so that it's useful for water quality managers. All right, so my mean foo is not great, but I took this one. Obviously, if you're going to have these long-term data and you want them to be useful, you need some kind of monitoring program would be the first assumption. You have to have a long-running monitoring program so that you can say something about what things were like before compared to what they're like now. And indeed, there are some monitoring programs out there that have been running for a very long time. Around the oceans around Belgium and the west coasts of European countries, Chesapeake Bay program, the National Aquatic Resource Surveys, the National Rivers and Streams Assessment has been running for 12 years. So we have programs in place that have been monitoring condition, including algae and phytoplankton as part of their process to track long-term change. And I've been extremely fortunate to be involved as a PI in collaboration as a collaborator with the US EPA's Region 5 office on the EPA's biological monitoring program throughout the Great Lakes. This is a monitoring program that's been running for 36 years, where the whole system is sampled twice a year in the spring and in the summer. I know many of you who are on the line here are well aware of this program. As part of this program, there's collection of water quality, zooplankton, phytoplankton, invertebrates, sediments, and all sorts of other contemporary studies that could take place where people can jump on board the research vessel Lake Guardian and do additional studies. This was initially established to comply with requirements under the Great Lakes Water Quality Act of 1978, largely driven by a lot of the well-known problems in Lake Erie and some in Lake Ontario where people suddenly realized, hey, we need to do something about this. We need to be tracking what's going on in the Great Lakes system. Again, a number of people are very familiar with this suite of sample locations. These are the sample locations that are sampled twice a year, in our case for phytoplankton assemblages throughout the Great Lakes system. This is the rosette sampler that is lowered down the water column to collect integrated samples. A little more detail, we also sample the deep chlorophyll layer, and we've now expanded this work from not just these deep pelagic areas to near shore as well. There it is lowered just below the surface. And again, what I'm talking about today are these algal assemblages. For the most part in the open water, we're talking about the phytoplankton, the soft algae and the diatoms and so on living out in the open water of this system. So this program has been great in that it has run long enough that we have been able to see some of these long-running changes that have taken place. These are just some example data. We have newer data than this. I'm trying to use data that are already in published reports, and you'll see that where I can, I'm providing the reports. And when I get to the end of my talk, I'll have a list and a place you can go to download all of these reports in case you're interested in more details of what I'm presenting. This is just a case here. We can see quite clearly the recent rise in the abundance of algae in the central basin of Lake Erie. This is something that has become quite well known related to the Western Basin harmful algal blooms that we've been seeing. Converse to this, we've had this invasion and proliferation of the non-native quagga mussel throughout the Great Lakes system, particularly in Lake Michigan and in Lake Huron. And we see this so-called collapse of the pelagic system early in the turn of the 21st century, particularly where we see the loss of these diatoms, these things that are easily filter fed and sink quite rapidly. And since then, the abundance of algae out in the open water has not recovered. So these are some of these data that we've been getting, quite useful data from our existing monitoring program. And I want to say in general, if we're talking about world systems, monitoring programs, as great as they are and as great as the Great Lakes program is, we're missing a lot of data about lake systems worldwide. In fact, fewer than 0.1% of the world's lakes are actually being monitored in any way. Funding obviously is a challenge. I'm sure everyone online is well aware of funding challenges with keeping long-term scientific programs running. I'm going to get into a bit later when I talk about QA, but taxonomy needs to be a concern. Algae, phytoplankton are just brutal in terms of their complexity. And if we want to maintain long-running monitoring programs for something like phytoplankton, we need to ensure taxonomic harmony over time, especially if the analysts are changing. And I'm going to talk a bit more about that later. And of course, we're missing all the data from before these things started. I mean, we obviously don't know what algae were living in the system 300 years ago without some mechanism to go back and examine pre-impact conditions, which would give us a context for remediation of these systems. Not to mention, they're all, monitoring data can be sporadic. They tend to be based on synoptic or snapshot measurements. So, for instance, in the Great Lakes, we are missing so much of what's going on in the wintertime under the ice, which undoubtedly is quite important to the ecology of the system. So, thankfully, as part of the EPA program, we were permitted to collect sediment cores from throughout the Great Lakes, and we added a paleolimnological program. And this allowed us to go back further in time. And again, I know some of you online, I'm preaching to the choir here, but if we take this plot again of our monitoring data, in which case we were lucky enough to go back to the 1970s, we paleoecologists, we flip these upright like we would have in a sediment core record. The top would represent recent records, the bottom would be very old, hundreds or thousands of years old. So we flip them upright so that we have a vertical temporal record. And in the case of paleolimnology, it allows us to extend our record farther back in time. It could be thousands of years in the case of the Great Lakes system, which is approximately 11,000 years old. As long as we can find sediment records that go back that far, we can do a reconstruction going back that far. If we're looking at older lakes that have not been glaciated in recent centuries, we could potentially go back millions of years. So if we think about a smaller lake, for example, pretend this is the same lake 220 years ago versus today, there are all kinds of questions out there about what has taken place over time in terms of supporting management of this system. First of all, there might be anecdotal data that out there or monitored data that it is a damaged system. Is it impacted? Well, the fossil record can tell us in terms of the fossil record, is it impacted? What are the causes or the potential causes of that change? When did it start? If we can determine when it started, we can pinpoint causes. How bad is the situation? And when it comes to recent times, is remediation actually working? Are we seeing, based on this fossil record, that things are getting better, maybe going back to pre-European settlement conditions or something better? And this last one, what can we expect in the future, is something even in paleolimnology studies has been, for the most part, ignored, asking this bigger question about what's going on in the recent fossil record that can tell us about future conditions, so we can be warned about what it might look like in the future so that we can start to manage the system now. So it's a muddy job. Again, I need to update these photos. It's quite an old photo of me, but we collect these sediment cores, and the cores contain all kinds of information. I'm going to talk about diatoms here for the most part, but there's all kinds of material coming from external sources, whether they are atmospheric contaminants or pollen from the terrestrial system, autochthonous indicators from within the system, the algae, the zooplankton, the fish. There's all kinds of DNA that end up depositing in this sedimentary record over time, thereby allowing us to reconstruct long-term records based on what sorts of stressors we're interested in. Even in the simplest way, you can see a lot of these cores have color records of what's going on. We can see in this case when human development began, an increase in organic material. We can see an intense storm event, for example, where a lot of inorganic material came in, and then this black, highly organic recent record of human development. We push these sediments up and extrude them into samples, which we can then analyze. And so it should be intuitive by now that these recent sediments are telling us about very recent times. The upper centimeter might represent a year or a couple of years. In this case, this is a core from Minnesota, in which case the bottom sediments represent about 250 years ago. If this length of core came from Lake Superior, this would be well over 1,000 years of sediment record. And as I move into explaining some of our results, we're going to focus on the diatoms. Now, these diatoms are the siliceous algae. They have biogenic glass cell walls, which makes them great as fossil bioindicators because they are resistant to desiccation and dissolution, and they tend to be preserved for thousands or to millions of years in these sedimentary records, thereby giving us great long-term indicators of change. All right, so getting into some of our project results, I'm going to provide 3 fairly quick examples of ways we've used these fossil records to provide early warnings about what's going on relative to climate change, eutrophication, and invasive species. All right, and this is some work that has been published in Limnology and Oceanography. Let's not forget that the Great Lakes represent almost 90% of the surface freshwater resource on the continent. And I think by that nature alone, it makes it an extremely important system to preserve and to ensure we're protecting in the future. And these results were the first that I know of to actually show a biological response to climate change in the Great Lakes system. This is a species called Pantoxichiella laurentiana. This was just named by Elizabeth Alexson last year. This group of organisms we call the cyclotelloid, or the circular diatoms that live in the phytoplankton in the Great Lakes, have become the poster child for climate change in the phytoplankton and freshwater systems. This is actually pretty amazing because this species is, in the late summer, one of the most abundant species in the Great Lakes, making up easily more than the biomass of all the fish in the lakes, and yet it wasn't even named until last year. We are seeing these organisms in the late summer in our monitoring data. Presumably, it seems, as a result of changing water condition. We're seeing them change in the upper records of these sediment cores. Now, we know the lakes are warming up. These are some plots of the recent surface water temperature data as published in Limnology and Oceanography. We know all the lakes are warming up quite rapidly. Just to use an example core, this is one core we collected from Eastern Lake Superior. Here's Kitty Kennedy. In this case, the core was collected using this box core shown on the left, and then we subcored it using tubes. We extrude these sediments into containers and do these analyses for the diatom records. And just a quick look at some of the dominant diatoms that we see through time. Again, bottom is old, top is recent. Pre-European or pre-impact diatom assemblages look like this, and you can see distinctly the dominant species changed fairly dramatically over time into recent times. In more recent times, many more of these so-called cyclotelloid, what we call Cyclotella sensulato, they all used to belong to the genus Cyclotella, and they have more recently been split into a number of other genera. So I will more colloquially use the term Cyclotella to describe these species that seem to be more abundant in the modern sediments. Here's a look at all of these cores that we collected through the Great Lakes system, indicated by different colors. And you see, no matter where you are in the Great Lakes, and despite all of the other things that are going on throughout the Great Lakes, we know that each Great Lake has a unique suite of stressors that we need to deal with. But it doesn't matter where you are, you're seeing an increase in these Cyclotella in the sediment record, in the relative density of the diatom species. And so whenever you correlate this with the available long-term data that we have for the Great Lakes, we see that the one thing that consistently in every lake, no matter where we are in the Great Lakes, is we see a correlation with the annual minimum temperature. The one thing that seems to determine the length of the ice-free season, how thick the ice is, other things like the maximum temperature does not seem to really be changing in the surface waters of the Great Lakes. The minimum seems to be changing the most. And we see it having the strongest correlation to our relative abundance of these cyclotelloid diatoms throughout the Great Lakes, providing strong evidence that it is related to the changing climate that is probably increasing these ice-free times. Currently, we don't have a great idea of the mechanism. It is very likely due to the physical factors that are driving stratification. We're having a longer stratified period later into the summer and fall seasons, which supports a longer period for the Cyclotella diatoms to thrive. Now, who really cares about Cyclotella? They're not really a species of concern. They're not invasive in the Great Lakes. They aren't clogging intakes or anything like that, but they do, like I say, make up an important part of the base of the food web. And it's likely we're not tracking everything that's changing as a result of climate change in the Great Lakes. We're just seeing this one group of diatoms that seems to be changing. There are probably other things changing as well. So If we think about the shift in the phytoplankton to these more small-celled organisms versus what was there and what was more abundant 30 to 40 years ago, these larger, juicy diatoms that are food for larger zooplankton and so on. This change from the left to the right to these new species is maybe something we should be concerned about, because there's no doubt the organisms that feed on these primary producers are changing as well. It's just something we haven't traced yet. So that is our climate early warning. We are seeing this change to a quite different assemblage. In all of the Great Lakes, the phytoplankton assemblage is nothing like it was in pre-European times, and there's no going back because of this warming and all of the other stressors throughout the Great Lakes system. So the fact that we've seen this first base of the food web change in the phytoplankton should serve as a warning that we are seeing these biological changes. I want to move on to the nearshore in the Great Lakes system, and this is a recently completed project on the St. Louis River Estuary area of concern, and how we've used paleolimnology to support management in a nearshore system. This is the area of concern surrounded by the red line, which is a portion of the catchment of Western Lake Superior that feeds into Duluth Superior Harbor. This area of concern was defined in the late 1980s. One of the beneficial use impairments was excess loading of nutrients and sediments. I'm going to focus on the discussion of the nutrient part of it here. And even though we have some good long-term monitoring data in the main stem of the St. Louis River Estuary for the nutrients to track change, up until this time, we didn't really have a good idea of how any remedial measures might be positively affecting the ecology of the system. We needed information to confirm things were actually better so that we could remove this beneficial use impairment and move toward delisting of this area of concern. So we would expect things to be better, right? We now have state-of-the-art sewage treatment in Superior, Wisconsin, Duluth, Minnesota. We know that the effluent they're sending out to the system is much better than it used to be in the early 1970s. So presumably the ecosystem should be responding and should help us to support the delisting of this AOC. So we went out and cored this system, this time through the ice. Paleolimnology, I prefer to do this coring on the ice because it provides a nice stable surface to work from. We do it from boats as well, but I prefer wintertime field work. The red dots on this map show the core locations, and in fact, the far western Lake Superior also is contained within this AOC. So examining change in western Lake Superior was also an important part of this, as well as the Thalweg and the bays throughout the estuary. And the purple dot shows the location of where monitoring has been taking place as well for phosphorus and other nutrients. All right, so here's a complex set of data. This is not something that I would expect to appeal to everybody, certainly not to water managers. Those of us who are the diatom nerds can look at this and can get some information from this because we know the species and we have at least a vague understanding of what they mean. We see on the left is the temporal record. The sediments are dated using lead-210 to provide time for all of our samples throughout our sediment record across the top of the species, and you can see as you go from bottom to top, you can see how things changed over time. For instance, we see a rise and fall of certain taxa. These are taxa that we know are higher nutrient species in Western Lake Superior. And again, this rise in Cyclotella, again, there's a climate story here, but these Cyclotella are also very low nutrient diatoms that reflect a lower nutrient ecosystem. And that's fine for those of us who actually do these counts and look at these data, but again, taking this and giving it to a water quality manager, maybe not so useful. We need to distill this down into something quantitative and useful, and we can do that because every diatom species has an optimum and a tolerance across a variety of environmental variables, in which it prefers to live. So this particular species, such as this pennate diatom, as an optimum where it would tend to occur at highest relative abundance, and a range or a tolerance at which it would prefer to live. So if we think of this environmental variable as something like phosphorus concentration. And we have calibrated our diatoms throughout the Great Lakes using training sets. So we have, again, a suite of phytoplankton collections and corresponding water quality data for all of the Great Lakes. And as part of an earlier program, the Great Lakes Environmental Indicators Program, which was had a coastal focus throughout the Great Lakes system, we have onshore collections of these diatoms and corresponding water quality data. The great thing about having these two matrices of data is that we can go in addition to the detailed water quality data, and here's a picture of me about 15 years ago when I used to actually sit at the microscope and analyze these. We have our complex set of diatom data to go with it. We can take those two matrices, put them together, and calibrate our diatom species to determine what their environmental responses are across these environmental gradients, such as phosphorus. This example shows 5 species, but in the Great Lakes, we are actually dealing with hundreds and hundreds of species. So we have hundreds of tools at our disposal that we can combine to infer condition. So now if we take fossil diatom assemblages from our sediment cores containing those diatom assemblages from the past, plug them into our diatom-based transfer function, okay? So these are all of our species coefficients to a variable like phosphorus. Based on the weighted average abundances of the species in our sediment samples, we should be able to infer past phosphorus concentrations in the system. And that is something that we have done for Lake Superior. And again, some of the things I've been talking about are kind of bad news stories, but there are good news stories as well. This is a case where we see the rise in the 20th century of phosphorus concentration in the system. Not too surprisingly, we had urban development, we did have agricultural development, and as we move later in the century, we have the Clean Water Act, the removal of phosphates from detergents. There was an exodus of agricultural activity in the Lake Superior Basin. So we see this improvement or a reduction in diatom inferred phosphorus in recent times, indicating to us that things like sewage treatment were actually working to reduce the nutrient load within Lake Superior. So good news. But it wasn't all good news. If we move into the deeper part, the nearshore part of the AOC, such as Aloise Bay, anecdotal data will tell us that indeed we're seeing this as a very bloomy system. We've been out and sampled this a number of times. Sometimes it has a very heavy algal load. And when we look at the fossil record, we quite clearly see there is this grab bag of well-known high nutrient diatom species fairly persistently increasing near the top of the sediment core from Aloise Bay. Maybe not such a good news story. And when we plug some of these nearshore bays into our transfer function and get our inferred phosphorus, we see this rise from what is already a pretty productive system to a much more productive system, in some cases perhaps even a hypereutrophic system. So even though we're seeing some improvement in the deeper portions of this AOC, the nearshore portions are showing this fairly persistent rise in diatom inferred phosphorus in the system. And even though I don't have time to get into all the different types of fossil indicators we can use, we have so many different things at our disposal. We also, in these cores, these nearshore cores, we analyze for the fossil pigments. So it allows us to move beyond just the diatom records, but into the soft algae whose hard parts don't necessarily preserve in the fossil record, but their pigments do. They're photosynthetic pigments. And we see the red and the pink. These are the bay cores, and we see quite clearly near the surface of the core the rise in these different types of pigments. Perhaps the most concerning is the rise of this phanezophil and myxoxanthophil, which are specific pigments to cyanobacteria, suggesting that we might, like we're seeing elsewhere, be seeing a rise in the potential for harmful algal blooms in this system. We have not seen them in this system yet in the deep into the estuary here, but there is potential for it because we're seeing this rise. All right, and so this has obviously given us somewhat of an early warning of what's going on in this system, something we were not aware of previously. Again, we want to provide information to managers. How do we use these data in a useful way to predict the future? So if we have, again, these nutrient-loving diatoms on the left, how they've increased, the diatom inferred phosphorus in Aloes Bay and how it is increased, there are ways to project this farther into the future, I believe. We should be able to monitor these data. We might have to make some assumptions. Don't worry too much about the numbers. These are just fairly arbitrary projections that we've modeled to look at the ways things may look in the future if this trend continues based on the fossil record. If we assume that it's a linear scenario, by 2050 we might expect to break 100 micrograms per liter phosphorus in the system. If it's a log normal scenario, which would suggest some nutrient remediation, we might be looking at 71.5, something in between with some continued increase in inputs with some remediation. We might have some phosphorus value that's in between. Now, the warning is here. What we do with it, we can decide in discussion with water quality managers. I just present this as a possible scenario of ways to predict what the future will look like under future remedial scenarios. So ultimately for this nearshore system, in terms of supporting delisting of the area of concern, things are looking much better in Lake Superior. I didn't show the deep estuary cores, but indeed we saw improvements or the stabilization of nutrient loads there. But near shore, not so much. But indeed, we've been warned that there's clearly some sort of problem here. We are still moving forward with recommending delisting of the area of concern because whenever the remedial action plan for this AOC was in place, we didn't know about a lot of these other contemporary stressors in the system. In many ways, I think the remedial action plan is obsolete, and we need to come up with something new for remediation of this system, because whatever's driving this nearshore change, whether it's legacy sediment phosphorus, climate change, increase the number of storms and their size, the amount of precipitation, may also be feeding into these problems. These are things we need to figure out, and we need to plan for life after delisting in this system. And I just want to mention, these diatom records, this is not limited to the Great Lakes system at all. We see this repeatedly, and I know I'm throwing a lot of data at you here, but again, consistently in Lake of the Woods, another, some people would say a Great Lake, we've seen this loss of the pre-European species to be replaced by many of these nutrient-loving centric diatom species, and everywhere you look, it's hockey sticks again, much like the climate plots. We see these hockey stick shaped graphs in our fossil records repeatedly, and we see this very recent increase, which seems to go along with the anecdotal information about harmful algal blooms in Lake of the Woods as well. So we are not limited to the Great Lakes in this work at all. All right, and I want to just go talk about invasive species very briefly. Just one slide, in fact. This is some work. I've been collaborating with Nikki DeWeese and Don Brandstrader on this work, and there's some published work on this, that these fossil records contain much more than just diatoms and pigments and these things I've been talking about. We've got the remains of the zooplankton and even the hard parts remains of a number of these non-native species like Bithytrephes. This is from a sediment core in inland Minnesota. We have not done these sorts of examinations in the Great Lakes yet. But if you look, this is some extruded sediment with one of these Bithytrephes spines sticking up out of the sediment. And we've been able to look at the remains in the sedimentary records to show that indeed we're seeing this fairly persistent increase in the abundance of this non-native species in the sedimentary record. And we detected them well before they were actually detected in monitoring records, sometimes decades before they were picked up on fishermen's lines and so on. So there's a lot of information in this fossil record that we can use to tell us about the past dynamics of the non-native species that have invaded these systems. And I would bet there are a number of lakes, such as in the state of Minnesota, that we are not even aware have been invaded. And I think these sedimentary records could tell us a lot about how abundant these non-native species are in inland lakes, let alone the Great Lakes. So to make a recommendation out of this, I hope that I've shown that in addition to these monitoring systems that we have in place, the fossil record has a lot to offer. We can decide what these recent records, these upper parts of these sedimentary records, mean in terms of making future predictions, but they are very telling, and they provide quite a bit of information that supports what we may or may be finding in our monitoring records. In a case like this, whatever this indicator is it getting worse? Can it be stabilized? Can it get better with some remediation? But obviously, there's a warning in place here. And the shorter way of saying is be warned, get sediment cores. All right, so there. Now, I wanted to move on. How much time have I got? I've got a few minutes left here. I want to quickly talk about our the QA system we have in place. We have in terms of quality assessment, we have a lot going on. I want to talk about the major component of that whenever we deal with algae, phytoplankton, diatoms. These are complex assemblages. We have thousands of species in the Great Lakes. So there's a lot of room for error among analysts. A key part of our quality control and quality assessment in our Great Lakes program is to make sure different people can get the same answer. If I get hit by a bus tomorrow and someone else takes over the Great Lakes program, can someone else actually do that? Can they do the analysis of the phytoplankton assemblage and get the same answer as another analyst? So that's a big question because these data are so complex, and this is some work that's been led by Katya Kovalenko in our research group. So just to give you some idea of this problem, now the lighter colored symbols here these are NMDS scores, so assemblage scores of the assemblages that my research team has analyzed, and the darker symbols up here are symbols that were analyzed by a group that did these analyses prior to us taking a role in 2007. Now, whenever we do these scores, we would expect some overlap because these samples have come from the same lake systems. And we see this very clear divide. And this is really, really unfortunate because, first of all, between this time, 1996 and 2000, and then jumping to 2007, a lot went on there. Is this a result of the invasion of the quagga mussels, or is it related to harmful algal blooms? I would say looking at this, maybe, but there's no way of confirming it. It's very likely this division in the scores in our assemblages is due to analyst error because the analyses were done by two different sets of contractors. So this is a real problem. I would never recommend taking all of these data together and trying to do some long-term analysis of the condition in this system because we obviously have some kind of analyst artifact taking place here. So this just gives you some idea of the importance of making sure you harmonize your taxonomy if we're going to do these long-term assessments. This has been observed numerous times looking at algae data. This is not my work, but a diatom so-called ring test where a set of diatom slides are made from the same set of samples, and they're sent around the world to a number of labs, and they're allowed to just go do your analysis and send your count data back to us. Again, they did an ordination to score these data, and you see again, these are all supposed to be perfectly overlapping since they represent the same set of samples, and clearly this is not the case. Harmonization is critical because of the complexity in these data. Here are some random photos of Great Lakes samples collected by Elizabeth Alexson. You can see this is what they actually look like whenever we start doing soft algae analyses. They're pretty diverse. Since we've been doing the Great Lakes program, we have 235 samples where we have done dual analyses, and the requirement under EPA standards is to reanalyze at least 10% of samples by another analyst to check and make sure, like I say, they're getting the same answer, that those assemblages, those communities algal communities are matching up in those paired samples. We've done this 235 times using phytoplankton samples from throughout the Great Lakes, and we calculated dissimilarity, the Bray-Curtis dissimilarity, to tell us about whether they are similar enough, first of all, to pass expected criteria. And, you know, the most important question is, even if there is some dissimilarity in these counts, do they provide the same utility in terms of environmental inference? Okay, so if you look at some of these, there's certainly a lot of room for error. If one analyst does a count and they miss something like this giant dinoflagellate here, you know, in terms of biovolume, that can really influence the similarity between two samples. A lot of these species are quite small, and the smaller taxa in terms of biovolume will tend to make up less of the amount in that sample. And if you miss one thing that's big because you went off on a different transect than the other analyst, you know, these sorts of things can contribute to big changes. So let's actually look at the results of these data. Again, these are all of the comparisons we've done to date since 2007 on these samples. Looking at this, this is the Bray-Curtis dissimilarity. So we can see the dissimilarity relative to a suite of random dissimilarities among samples throughout the Great Lakes. Looks pretty good. This is the criterion. We expect 60% precision. This is what the US EPA requires, or at 0.4 Bray-Curtis dissimilarity. All right, so we can see for the most part, aside from a couple of stragglers, we're meeting these criteria in these analyses. We have a mean of 0.17, and most of them meet the criterion. So, you know, that helps me sleep at night. That tells us that we're meeting the criteria as expected by the US EPA. Not surprisingly, you know, if we just take a bunch of random comparisons of phytoplankton samples, in many cases we get close to 100% dissimilarity. That doesn't surprise me. But we just put this here for reference to show that indeed our QA comparisons are pretty low compared to what we would expect randomly. But the question is, first of all, what is driving the dissimilarity? And it's pretty obvious that the diversity of the samples is a major driver in this dissimilarity in the system. We think of something like, in this case, diatom assemblages. Very low diversity, a lot of similarity, a lot of things like these centric diatoms can sometimes dominate the samples versus something much more diverse in our prepared samples. As we increase the diversity, we see an increase in dissimilarity. We probably shouldn't be too surprised by that. The more diversity we have, the greater the chance there is for an analyst, you know, not necessarily making a mistake, but having a different perception of some of these what these rare species are compared to another analyst. So that's not unexpected. Looking at the seasonal effects, again, we have spring and summer collections. We see that the spring collections have a slightly higher dissimilarity overall. And that, again, doesn't surprise me because springtime tends to be more diverse by the summer. The nutrient limitations have really kicked in. There is a loss of a lot of the sensitive species that disappear throughout the summer, and lower diversity, not surprisingly, lower dissimilarity in the summertime. So season matters. But like I say, what does this matter in terms of actual inference of information? There might be some dissimilarity. In worst case scenario, if we have a 40% dissimilarity between two samples, if it still gives us the same answer in terms of environmental information that is important for management, maybe it's not that big a deal that we have what seems to be, you know, not a very desirable dissimilarity. 40% dissimilarity for someone analyzing zooplankton or fish probably doesn't sound very good. Is it okay for phytoplankton? Until now, it's been kind of an open question. So we can actually generate inferred information that might be useful to managers in the form of algae-inferred phosphorus concentration. So if, again, we take this algal assemblages, plug it into our algal-based diatom or algae transfer function, spit out inferred information, what happens? And here we have a pairwise comparison of these QA samples, again, the primary and the QA samples compared. And indeed, we see based on the gradient of phosphorus throughout the Great Lakes system, the ideal would be everything would fall right on the one-to-one line throughout the system. We see there is some scatter around this, but look at this, it looks okay. It looks like even though there is some dissimilarity, we are generally getting the same answer in our inferred phosphorus. Does poor similarity actually result in poor inferred water quality? Well, within the range of dissimilarity that we're seeing in the samples, generally no. We see one rise in our Lake Huron samples, largely driven by one dissimilarity that is outside what we would prefer to be outside this 40% dissimilarity. So that may be driving this. There was no actual significant change in our inferred values within the range of dissimilarity that is allowable. So this is good news to me. So in conclusion, based on these assessments, again, we're just getting started in these analyses, but first of all, we're largely meeting our acceptability standards for phytoplankton assessments in the Great Lakes work. It appears that diversity is a substantial driver of the amount of difference we see in comparative sample analyses. And within the acceptable range, we are seeing no real effect on the performance, at least in terms of phosphorus models. Now, we need to continue this work and further determine whether 40% dissimilarity is really okay. There are other things like, if we look at the sensitivity of the biomass change over time, we want to be able to sensitively detect historical changes in our monitoring data or in our fossil data that can tell us about changes in the abundance of the algae relative to harmful algal blooms. If an error of something like 40% is too blunt to actually detect the changes we want to see, then that's a real problem. And so that's something that we are continuing to analyze now, and there'll be more answers on that in the future. So I wanted to wrap up. Hopefully there is, for some of you, there's a new respect for the fossil record. I think that there's a lot more we can do with fossil records in combination with ongoing monitoring programs in the Great Lakes to support management of deep water and coastal systems. If you want to download this PowerPoint and all of the published papers that I've cited, I've provided a bit.ly link here if you want that. And I'm going to end it there. Thanks a lot.
Ewan, thank you so much. Wow, that was fascinating. I'd like to open it now to turn it over to those that are attending to ask any questions. I'll turn it over to Elizabeth. Can you join us? Are there any questions that have come in?
Thanks, Craig. Not yet. I do want to mention, folks, I have posted this link that you see on the slide here. I have posted this link into the chat function so that you have access to directly download the PowerPoint and cited papers. If you have any questions, please type the question into the question box and I will read them out. You may also raise your hand and I can unmute folks one at a time. And we have, okay, hold on here. We got a couple questions coming in. The first question by Ellie Hagan. Do you know how the 40% precision QA requirement was determined by the US EPA?
Right, so that this is something that was determined before my time. As I recall, it was determined because that was the average similarity that they were getting in the average dissimilarity they were getting in these analyses from the 1990s, and they set it as what seemed to be reasonable or realistic. We didn't know whether it was, I don't think they knew whether it was meaningful at the time, but it seemed realistic at the time. So until now, it's been fairly arbitrary.
Okay, and I'm going to unmute Tim Lewis, who has his hand raised. Tim, you're open.
All right, can you hear me?
Yes.
I can.
All right, great. We kind of dealt with this same problem when I was at the Army Corps of Engineers with the quality assurance of samples, primarily looking at veligers and the quantity of veligers in a water sample. And we're starting to look at a comparison between looking at basic microscope counts and then also a device called a flow cam. Are you familiar with that?
What is it called again?
Flow cam.
Oh, flow cam. Yes, absolutely.
Yeah, you pass your sample through a flow cam and it takes photographs of all the different phytoplankton and zooplankton and veligers and everything else. And then you can develop a photo library that will allow you to do a comparison across the board for different samples. I was wondering whether you've considered going that route for maybe improving on the reproducibility of the analysts across the board.
Yeah, so I'm very familiar with FlowCam and what it can do, and we've considered it for use in some of our ballast water programs. In this program, the taxonomic requirements are too fine for FlowCam to be useful in terms of identification. So we haven't gone that direction because the FlowCam photo quality is not good enough to give us the taxonomic information we need. It may be used for something more general like density and general biovolume measurements, but whenever taxonomy is involved, not so much here.
Okay. And one more question related to the change in composition of your assemblages. Has anybody been, I know they've done some of this work earlier with zebra mussels and looking at bioenergetic modeling. Have you been able to say whether the change in assemblages and the plankton out there might become more conducive to the establishment of Asian carp in the Great Lakes?
I have not. Yeah, I have no answer for you on that one.
Okay. I just wondered.
Sorry.
I'm not sure exactly what they eat, I'm not sure what they like to eat, but just wondering.
Okay, thank you, Tim. We've got a couple other questions. Lou Bloom, who is the QA manager for US EPA Great Lakes National Program Office, just said, great job, UN. He appreciates the ending linkage to quality control at the end.
Thanks, Lou.
We've got a question from Liz Favot. Great talk, UN. Is there a way to reconcile or incorporate increasing low TP optima diatoms or small cyclotellaloids, sorry, I don't know how to say that are increasing because of climate-driven changes in thermal stability and potentially making diatom inferred TP appear as if it is decreasing when in fact it may be stable?
Yeah, so that's all part of the whenever we develop these diatom-based transfer functions, we need to be very careful to make sure that what we're inferring is actually the variable we are interested in. And so, yeah, in the Great Lakes, we have this, in fact, the climate effect, I would say, is very much related to changes in these open water nutrients. So the diatoms probably aren't directly responding to climate in any way. They're responding to changes in the system. So what we're probably seeing is truly a nutrient signal in the open water because of the stratification that is resulting in lower and lower nutrient concentrations in these pelagic systems. But without getting into too much of the complexity, there are a number of systems in place to confirm when you do an inference, is your fossil record really providing realistic inferences of the variable you're interested in? You need to confirm that your species are responding to your variable of interest, and we can do that, including for these centrics. I think getting into those details might be a bit much for me to talk about here.
Thank you. We've got another question coming in from Jan Krakowski. What does the future hold in terms of water quality for the Great Lakes? Any increase in HAB cyanobacteria and what future management mitigation could be taken?
Yeah, good question. Not being a policymaker, I can't really give you a clear answer on that, but we've seen repeatedly, both in monitoring data and these fossil records, we see a rise in the potential for harmful algal blooms. We're now seeing more of these coastal blooms taking place, so it's certainly something that is getting worse. In terms of recommendations, the first exactly is causing it. We don't truly know that at this point. It's more than just what we see going on in Western Lake Erie. There's more than agriculture. There seems to be a climate effect. There seems to be a storm effect, but it's something that needs to be sorted out. At this point, my hope is that we can recommend for agencies and policymakers that we need the funding to actually do the work to sort out what this problem is and where things are going in the future. So we can make recommendations that are suited to each location where there's a concern.
We are at the top of the hour. Craig, can we keep going? We've got 2 more questions.
It's okay for me.
I'll just keep rolling with it then. Okay, so we've got a question by Maria de Los Angeles Gonzalez Sagrario. Have you tried to detect early warning signals of regime shifts? For example, have you considered changes on resilience indicators like standard deviation, autocorrelation, return rate? If so, how was your experience?
Yeah, so I haven't done that. There are a lot of other aspects of these early warning signals, including like regime change, often which is very sudden. In my experience, they seem to be these very gradual changes leading to something new. If you think of that in geological terms, it might be considered a very rapid regime change. Other cases, people have proposed analysis of flickering. I worry a bit about the resolution to actually detect flickering signals. Some of you may be aware that early warning signals often come in this form of flickering. You could have one bad season or one bad year and things go back to normal, and then another sudden bad thing happens. This periodic change ultimately resulting in a permanent shift to an undesirable state. That is not something I've worked on. It's not something I've seen in my fossil records, but it has been theorized quite a bit in the literature for these records.
We have another question from Heidi Rantala asking, is there evidence of changing the algal community related to top-down effects of spiny water flea in Nikki's project? If so, can you differentiate those changes from changes related to climate change?
We're still writing it. Let's see. Wow, I wish I had more clear answers for you guys. This is something we're actively analyzing right now. There does appear to be a relationship between, some relationship between the invasion of Dithotrephes and the response of its prey assemblages. The relationship to climate change and other prevailing variables around these lakes, we're in the middle of analyzing that. So again, I don't have a clear answer beyond that.
We have another question come in from Tom Hollenhorst. Could you discuss the potential of using in situ optical measures of pigments and particle size measured by towed sensor arrays, buoys, or AUVs as part of an early warning system? How best can that be integrated with taxonomy?
Yeah, so as long as that's, and this is being done, some in the Great Lakes. It's a shame we didn't start it 30 years ago, but so that we understand what those pigment signals from the water mean, we should be collecting phytoplankton samples along with it and looking at the correspondence between the actual communities and the signals we're getting from these towed devices. And I think we should be able to build tools that are quite acute in telling us about changes in the primary producer system in the Great Lakes.
Okay.
Okay, I think if we can have questions, you could contact Dr. Ewan directly. We appreciate it. I think we'll call it now. And you'll see on your screen, you'll see our upcoming webinars for the committee. Next month, Jeff King from the Army Corps of Engineers will be discussing their work with engineering using nature to help with the ecological restoration activities in engineering. In February, we have Doug Pearsall from the Nature Conservancy, and he'll be talking about coastal restoration and some of the making the case of benefits for people and does it matter. And then we also, in March, we have our presentation from some Canadians from the Central Lake Ontario Conservation Authority, Dan Moore and Heather Pankhurst, who have been using some of the coastal wetland monitoring data to look at the effectiveness of their restoration actions. So I think we have a number of very interesting webinars and invite you all to those and upcoming. Also wanted to mention there are some upcoming meetings and that the committee is being involved in. The Great Lakes chapter of the Society for Ecological Restoration, the Quebec RE3 Cubed Conference, might say, that will be held in Canada, in Quebec in June, and the National Conference in Ecosystem Restoration, which will be in Portland, Oregon. So we want to thank you all for participating and certainly appreciate Ewan for a very fascinating and very informative webinar. We certainly appreciate all the effort you went to put it together for us. Thanks again.
For having me.
Thank you, everyone. This ends today's webinar.